Alright, let's go. Number 10. The lodger in every one of your cells. Inside almost every cell in your body, there is a structure with its own DNA, its own double membrane, and its own reproductive schedule that runs on a timetable independent of the rest of you. It is called the mitochondrion, and there are somewhere between a few hundred and a few thousand of them in a typical human cell, more in muscle and heart tissue, which burn the most fuel. In 1967, the biologist Lynn Margulis published a paper that most journals had already rejected. Her argument was that mitochondria are not human inventions at all. They are the descendants of free-living bacteria engulfed by a larger cell roughly 1.5 to 2 billion years ago that were never digested. Instead of being broken down, they were kept, and the arrangement became permanent. The evidence was circumstantial at first and became overwhelming later. Mitochondrial DNA is circular, like bacterial DNA, not packaged into chromosomes the way the rest of your genome is. Mitochondria divide by splitting in two, the same way bacteria do, on their own schedule, not synchronized to when the host cell divides. Their inner membrane carries proteins closely related to those found in a specific group of bacteria, called alpha proteobacteria, a relationship confirmed by genetic sequencing in the 1970s and 80s once the technology existed to check. Margulis called the arrangement endosymbiosis, one organism living permanently inside another for mutual benefit. And by the time she died in 2011, it had gone from a rejected paper to a textbook fact taught in every introductory biology course on Earth. The bacterium got a permanent home with a stable supply of nutrients. The host cell got something it could not previously do efficiently: burn oxygen to produce enormous amounts of usable energy. That single arrangement struck sometime before the first animals, the first plants, the first fungi, is the reason complex multicellular life was possible at all. Every calorie you have ever burned to lift a hand or think a thought passed through a machine that used to be a separate creature. If you enjoy videos that reveal the hidden side of science and the incredible story of human evolution, hit subscribe now. We create documentaries packed with fascinating facts you won't hear anywhere else. Now, let's uncover the next secret. The genome tells the rest of the story. Over billions of years, the mitochondrion has lost almost all of its own DNA, handing most of its genes over to the nucleus of the host cell for safekeeping, until only a small, stripped-down set remains, usually 37 genes in a human mitochondrion, down from what would once have been thousands in a free-living bacterium. It cannot survive outside your cells. It cannot be cultured on its own. It is entirely dependent on proteins manufactured by your own DNA and shipped in. The merger, once it happened, became irreversible. There is a practical inheritance pattern that follows directly from this history. Mitochondrial DNA passes almost exclusively from mother to child because the sperm's mitochondria are typically destroyed after fertilization, which means the mitochondria in your cells right now are a direct, unbroken maternal line stretching back through every mother before your mother, all the way to a single population in deep prehistory. Population geneticists use exactly this fact to trace human migration because mitochondrial DNA mutates at a roughly predictable rate and never recombines with a father's line to muddy the signal. The number of mitochondria per cell is not fixed, and the variation tells its own story. A liver cell might carry a few hundred. A single muscle fiber in the heart, which never stops working for as long as a person is alive, can carry several thousand, packed densely between the contractile fibers. Because cardiac tissue burns through energy at a rate few other tissues in the body approach. Cells with almost no ongoing energy demand, like mature red blood cells, discard their mitochondria entirely during development and run without them. A reminder that the arrangement, however ancient and however permanent, is still tuned cell by cell to the amount of work being done. What is inside your cells is not, strictly speaking, entirely you. It is a captured bacterium that stopped being free 2 billion years ago and has been quietly running your metabolism ever since. Number 9. The virus that built your placenta. About 8% of the human genome is made up of sequences that did not originate in humans at all. They are endogenous retroviruses, the fossilized remains of ancient viral infections that inserted themselves into the DNA of a germline cell, an egg or a sperm precursor, millions of years ago, and were then copied faithfully into every descendant of that individual ever since. Most of these sequences are broken, disabled by mutations over deep time, inert junk sitting silently in your chromosomes. A handful are not broken. A handful still make protein, and one of them makes something your body cannot do without. In 2000, two independent research teams, one led by the French virologist Thierry Heidman, and one working at Genset in France, identified a gene embedded in human chromosome 7 that produces a protein called syncytin. The gene is not a human invention. It is the repurposed envelope gene of a retrovirus that infected a primate ancestor at least 25 million years ago. In a normal retroviral infection, the envelope protein's job is to fuse the virus to a host cell membrane so it can inject its genetic material. In the human placenta, that exact fusogenic property was hijacked for a different purpose entirely, fusing placental cells together into a single continuous barrier layer. The syncytiotropoblast that separates a mother's bloodstream from her fetuses while still allowing nutrients and oxygen to cross, blocks syncytin in a mouse model, and the placenta fails to form properly. The pups do not survive. This is not a decorative leftover. It is load-bearing, and it gets stranger under comparison because different lineages of placental mammals appear to have independently domesticated different retroviruses for the identical job. Mice use a different endogenous retroviral gene, syncytin A, acquired from a separate ancient infection. Cats, rabbits, and other mammal groups each carry their own independently captured version. Evolutionary biologists now describe this as one of the clearest known cases of convergent domestication, the same viral trick borrowed from unrelated infections, over and over, because it happened to solve the same problem, building a barrier that fuses cells into a sheet. The rest of the endogenous retroviral genome does not sit idle either, even where it makes no functional protein. Long stretches of these ancient viral sequences carry their own regulatory switches, promoters and enhancers left over from when they needed to control their own replication. And modern human genes have, in a number of documented cases, been rewired to run off those borrowed switches instead of building new ones. The virus did not just leave debris behind. Parts of its old control panel got wired into yours. There is a reason retroviruses are able to do this, and other viruses generally are not. A retrovirus carries an enzyme, reverse transcriptase, that copies its RNA genome back into DNA and inserts that DNA directly into the host chromosome. If that insertion happens to land in a sperm or egg cell rather than an ordinary body cell, the insertion becomes heritable. It stops being an infection and starts being ancestry. Every human alive is, in this narrow and literal sense, carrying the genomic residue of an outbreak that has no living victims left to remember it, because the virus is not out there anymore. It is not a threat. It is a fossil, and one very small piece of it built the organ that fed you before you were born. Number 8. The gene that stopped working and never had to work again. Almost every mammal on Earth can manufacture its own vitamin C. A goat under metabolic stress can synthesize several grams of it a day in its liver. No diet required. Humans cannot. Neither can other apes, nor old world monkeys, nor, oddly, guinea pigs, nor a handful of fruit-eating bats. Humans need vitamin C from food, and if we do not get it, the result is scurvy, a disease of failing collagen that killed sailors by the tens of thousands across the age of transoceanic travel before anyone understood why citrus fruit prevented it. The reason is written directly into the genome, and it is not a missing gene, it is a broken one. The last step of vitamin C synthesis in mammals depends on an enzyme called L-gulonolactone oxidase, GULO for short. Humans still carry the gene for it. It sits on chromosome 8, in roughly the genomic location you would expect given its position in other mammals, and it is unmistakably related to the working version found in species that still make their own vitamin C. It is simply riddled with disabling mutations, several of them independently capable of destroying the enzyme's function, accumulated over millions of years with nothing to stop them, because a mutation in a gene you are not using confers no disadvantage and therefore is not selected against. In 1994, the Japanese biochemist Muramitsu Nishikimi and colleagues sequenced the human version and confirmed what comparative anatomy had already implied. The pseudogene, GULOP, is a corpse of a gene, not an absence of one. The timing of the break is where it gets genuinely interesting. Comparative genomic work across primates places the disabling mutations in a common ancestor of humans, chimpanzees, gorillas, orangutans, and Old World monkeys, meaning the gene died once, in a single lineage, somewhere on the order of 60 to 70 million years ago, and every descendant of that lineage has been carrying the broken copy ever since. It did not have to be fatal. It only had to happen in a population whose diet already supplied enough vitamin C from fruit that natural selection had nothing to correct. A mutation with no consequence is invisible to evolution. It simply rides along. The guinea pig case, worked out independently, shows the same failure happening a second, unrelated time in an entirely different lineage. Evidence that this particular gene is a soft target, vulnerable to loss whenever a species' ecology makes the enzyme redundant. Fruit bats lost it a third time, separately again. The pattern that emerges is not one broken gene, but a recurring evolutionary event. Whenever a diet reliably supplies a nutrient, the machinery for making that nutrient becomes optional, and optional machinery, given enough time, breaks. You are carrying a fossil of a metabolic pathway your ancestor abandoned in the treetops, tens of millions of years before anything recognizably human existed, back when a diet heavy in fruit made the enzyme unnecessary. The gene is still there, in every cell, still recognizably related to the one a goat uses, permanently switched off, a record of a decision your body made so long ago that the species making it was not yet a primate as we would picture one. Number 7. The other human in your genome. In 2010, a team led by the Swedish geneticist Svante Pääbo at the Max Planck Institute for Evolutionary Anthropology published the first draft sequence of the Neanderthal genome, extracted from bone fragments recovered from Vindija Cave in Croatia. The comparison to living humans produced a result that overturned decades of assumption. Non-African populations today carry, on average, somewhere between 1 and 2% Neanderthal DNA, a fraction too consistent and too widely distributed across Europe and Asia to be explained by chance resemblance. It is inheritance. At some point, or more likely several points. After modern humans expanded out of Africa, they encountered Neanderthal populations already established across Europe and Western Asia, and the two groups had children together, repeatedly, over a span of thousands of years. The amount is small, but it is not evenly distributed across the genome, and where it clusters matters more than how much there is. Large stretches of the modern human genome are almost entirely free of Neanderthal ancestry. Deserts that suggest those regions were actively selected against, meaning offspring carrying Neanderthal versions of certain genes in those regions were, on average, less successful, and the sequences were gradually purged. Other regions show the opposite pattern, Neanderthal variants persisting at elevated frequency, which is the genomic signature of something that helped. The clearest cases involve the immune system. Work published by Janet Kelso, Michael Dannemann, and colleagues at the Max Planck Institute identified Neanderthal-derived variants in a cluster of genes called the OAS locus, and in the innate immune receptors known as toll-like receptors, TLR1, TLR6, and TLR10, at unusually high frequency in living Europeans. These genes are involved in the body's first line of defense against pathogens, and the inference is straightforward even if the mechanism is debated in its particulars. Neanderthals had lived in Eurasia for several 100,000 years before modern humans arrived and had presumably adapted, at the genetic level, to local pathogens that a recently arrived African population had never encountered. Interbreeding offered a shortcut. Instead of waiting for new mutations to arise and spread, incoming modern humans could acquire, in a single generation, immune variants that Neanderthals had already spent hundreds of millennia refining. Not everything inherited was a gift. A 2020 study led by Hugo Zeeberg and Svante Pääbo identified a Neanderthal-derived haplotype on chromosome 3 that is the single strongest known genetic risk factor for severe respiratory illness during the COVID-19 pandemic, carried by roughly 16% of people in Europe and up to half of people in parts of South Asia. The same immune system that borrowed useful defenses against ancient pathogens also inherited a liability against one that did not exist for another 40,000 years. Evolution cannot see forward. It only keeps what worked at the time. Skin and hair are marked by the same inheritance, Variants in the keratin gene cluster, inherited from Neanderthals, have been linked to skin tone and hair color in modern populations, and a 2014 study by Sriram Sankararaman, David Reich, and colleagues found that Neanderthal ancestry is significantly enriched in genes affecting keratin filament structure, the structural protein underlying skin, hair, and nails, suggesting the borrowed variants may have helped a recently arrived African population adapt to colder, lower light Eurasian environments faster than new mutations alone could have managed. Neanderthals themselves are gone. The last identifiable populations vanishing from the fossil and genetic record roughly 40,000 years ago, for reasons still argued over: competition, climate, absorption into the larger modern human population, or some combination running simultaneously. But absorption is not a metaphor in this case. It is the literal, measurable mechanism. There is no pure end to that species, because a percentage of it did not end. It is sitting in your immune system deciding right now how your body reacts to a threat that a species extinct for 40 millennia once had to survive first. Number 6. The gene that came from higher up and further east. Neanderthals were not the only extinct human lineage that modern humans met and mixed with, and the second group left a stranger, more geographically specific mark. In 2010, the same year the Neanderthal genome was published, a research team working with a single finger bone fragment recovered from Denisova cave in the Altai Mountains of Siberia sequenced a genome belonging to a population previously unknown to science. They had no complete skeleton, no clear picture of what these people looked like. What they had was DNA, distinct enough from both modern humans and Neanderthals to represent a third separate lineage. They were named, for lack of anything else to call them, Denisovans, after the cave. Comparison against living populations turned up something unexpected. Modern Melanesians and some indigenous Australian populations carry up to 4 to 6% Denisovan ancestry, the highest proportion found anywhere. Despite Denisova cave sitting thousands of kilometers to the north in Siberia, evidence of population movements and additional interbreeding events that happened somewhere in Asia that left no fossil trace at all, reconstructed entirely from the genetic signal carried in living descendants. The most striking inherited variant belongs to Tibetan populations living at extreme altitude. A gene called EPAS1 regulates how the body responds to low oxygen, and most humans at high elevation compensate by producing more red blood cells, a response that thickens the blood and, over a lifetime, carries real cardiovascular cost. Tibetans do something different. They maintain efficient oxygen use without the same overproduction of red blood cells, a genuine physiological advantage at altitude. And in 2014, a team led by Rasmus Nielsen and Emilia Huerta Sanchez traced the specific EPAS1 variant responsible directly to Denisovan ancestry. It is present in the Denisovan genome, essentially absent in most other human populations, and found at high frequency specifically in Tibetans, whose ancestors appear to have acquired it through interbreeding and then kept it, because in the oxygen-thin air above 4,000 meters, it worked. Nobody has ever found a Denisovan skeleton complete enough to say with confidence what the species looked like. A jawbone from the Tibetan plateau, a few teeth, fragments of skull from Siberia, and more recently, from a cave in Laos that is close to the entire physical fossil record for an entire branch of the human family tree. And yet their genetic signature is not a fragment. It is functioning, present-tense biology, currently allowing millions of people to breathe efficiently at altitudes that would strain almost anyone else's cardiovascular system. The species is known better through what it left in the DNA of people living on a plateau in Central Asia today than through anything recovered from the ground. Number 5. The reflex that used to move a coat, cold air, fear, a particular piece of music. Any of these can raise a fine stippling of small bumps across the skin, each bump centered on a single hair follicle, each one produced by a tiny bundle of smooth muscle called the erector pili contracting and pulling the hair upright. In humans, the reflex accomplishes almost nothing. Human body hair is too sparse and too fine to trap a meaningful layer of insulating air, so raising it does not warm anyone up in any measurable way, and it produces no visible change in most people beyond the texture of the skin itself. The muscle fires anyway, on a nervous system pathway that has not been switched off, The reflex is not a human invention. It is a mammalian one, and in almost every other mammal, it still does exactly what it evolved to do. A cat with its fur raised is trapping a thicker layer of insulating air against the skin, or, in a threat display, appearing larger to a predator or rival by increasing its apparent body size. Both functions depend on a coat dense enough to matter, and that is precisely the ingredient a hairless, or nearly hairless, primate lineage lost somewhere in its evolutionary history. Estimates for when human ancestors lost most of their body hair vary, but genetic evidence involving the MC1R gene and related pigmentation pathways, published by Alan Rogers and colleagues in 2004, places the shift to mostly hairless, darkly pigmented skin at over 1 million years ago, tied to the need to dissipate heat efficiently while walking and running long distances across open savanna. The insulating coat that made piloerection useful was given up for a cooling advantage. The reflex controlling that coat was not given up along with it. The wiring survived intact because the reflex costs almost nothing to keep, and because it is driven by the sympathetic nervous system, the same branch responsible for the broader fight or flight response, activated automatically alongside a racing heart and dilated pupils whenever something registers as a threat or a strong emotional jolt. Fear raises hair on a wolf for a defensive reason, to look larger, more formidable, less worth attacking. Fear raises the same muscles on a human arm for no functional reason whatsoever, an automatic broadcast to a coat of fur that stopped receiving the message millions of years ago, because the alarm system that triggers it was never rebuilt to check whether the equipment on the other end still exists. Every time skin prickles at a piece of music or a sudden chill, a muscle contracts in exact anatomical continuity with the one that once helped a furred ancestor look bigger to a predator, or trap a layer of warm air against a winter coat. The coat is gone. The reflex that managed it is still standing at attention, following orders issued to a body plan that no longer exists. Number 4. The muscle that only 3/4 of people still have. Make a fist and flex the wrist forward. And in most people, a narrow tendon will stand up prominently at the center of the wrist, flanked by 2 others. That tendon belongs to the palmaris longus, a long, thin muscle running from the inner elbow to the palm, and it is one of the most frequently cited examples of human anatomical variation taught in gross anatomy courses, because a substantial percentage of the population does not have it at all. Studies across different populations put the absence rate anywhere from roughly 10 to 25%, sometimes on one arm and not the other, with documented variation by population and by sex. Nobody who lacks it notices any loss of grip strength or wrist function, because 2 much larger muscles do essentially all the real work of flexing the wrist, and the palmaris longus, when present, contributes almost nothing to that job. Comparative anatomy makes the muscle's original purpose reasonably clear. In many other primates, and in mammals generally, the palmaris longus is well developed and connects into the palmar aponeurosis, the tough sheet of connective tissue across the palm, where it plays a genuine mechanical role in gripping, particularly in species that spend significant time climbing or hanging by their hands. A muscle that is small and functionally optional in a species walking upright on the ground looks very different in a species regularly supporting its full body weight from a branch. The strong working hypothesis, consistent with the pattern seen across the primate family tree, is that the palmaris longus is left over from an ancestor whose survival depended more directly on grip strength than any modern humans does, and that as bipedalism took over as the primary form of locomotion and the hands were freed for other tasks, the muscle's mechanical contribution became small enough that losing it entirely carried no cost, because it costs nothing. Natural selection has had no reason to preserve it, and the muscle has become one of the most variable structures in the human body, present, absent, doubled, or reduced to a thin cord, sometimes differing between the left and right arm of the same person. Surgeons take direct practical advantage of that redundancy, because the tendon is expendable and easily accessible. It is one of the most commonly harvested grafts in reconstructive surgery, routinely used to rebuild damaged ligaments and tendons elsewhere in the body. On the straightforward logic that a structure doing essentially nothing where it is can be repurposed to do something necessary somewhere else. What is unusual, is not that the muscle is sometimes missing. It is that so many people still have it at all, decades or millions of years past the point where it stopped being needed for the job it was built to do, quietly wired into roughly 3 out of every 4 human forearms, waiting for a climb that most of the people carrying it will never make. Number 3. The tail that never fully leaves. Every human embryo, at around the 5th or 6th week of development, has a tail. It is not a metaphor, and not a loose figure of speech used to describe something tail-like. It is an actual external structure, a genuine caudal projection extending beyond where the buttocks will eventually form, made of somewhere around 10 to 12 vertebrae at its peak, more than the final adult human spine will ever carry. For a short developmental window, a human embryo has a longer tail, proportionally, than some adult primates. The structure then regresses. Through a precisely timed process of programmed cell death, apoptosis, the tail's extra vertebrae are broken down and reabsorbed, and by around the 8th week the external projection is gone, leaving behind a small internal remnant fused at the base of the spine. The coccyx, the tailbone, typically 3 to 5 small vertebrae fused into a single triangular structure that has no vertebral discs, moves only slightly, and, on its own, bears essentially none of the body's weight while sitting, contrary to a persistent piece of folk anatomy. It still serves a real function, providing an attachment point for several pelvic floor muscles and ligaments that support the organs above it. But the function it serves has nothing to do with balance, counterweight, or grip, which were presumably the tail's original jobs in an ancestor that used it for exactly those things the way most other primates still do. The developmental regression does not always complete cleanly. In rare cases, the programmed cell death that is supposed to eliminate the embryonic tail fails to run its full course, and a child is born with a true vestigial human tail, a soft, boneless, or occasionally bony projection extending from the lower spine. Medical literature contains several dozen well-documented cases. It is not, contrary to the name sometimes used loosely in older or non-specialist accounts, a form of atavism from some other angle. It is understood as an incomplete regression of a structure every human embryo builds and then dismantles, a step in the standard developmental program that, in these specific cases, simply stalls partway through. The genetics behind the whole sequence, both the building and the dismantling, has become considerably clearer in recent years. In 2024, a team led by Bo Xia at New York University identified a specific transposable genetic element inserted into a gene called TBXT that is present in humans, chimpanzees, and other apes but absent in tailed monkeys. A mutation the study proposes played a direct role in the loss of the external tail across the ape lineage, roughly 20 to 25 million years ago, tested experimentally in mouse models where the equivalent insertion produced short-tailed or tailless offspring. The tail is not simply an old feature humans no longer have. It is a feature every human being still builds, on schedule, in the womb, before it is disassembled again on an equally precise schedule, and the fused, foreshortened evidence of that entire construction and demolition project sits permanently at the base of every human spine, whether anyone ever thinks about it or not. Number 2. The fold in the corner of the eye. Look closely at the inner corner of a human eye, where the upper and lower lids meet near the bridge of the nose, and there is a small, reddish-pink fold of tissue, semilunar in shape, sitting just beside the lacrimal caruncle, the fleshy pink node that produces some of the eye's oily secretions. It's called the plica semilunaris, and in humans it does very little. It has a small amount of smooth muscle inside it, allows for slight movement, and may play a marginal role in eye motion and drainage, but it is, functionally, a minor structure, easy to overlook, and rarely mentioned outside of ophthalmology and comparative anatomy texts. In a large number of other vertebrates, the equivalent structure is not minor at all. Birds, reptiles, and many mammals possess a fully developed nictitating membrane, a translucent or semi-transparent third eyelid that sweeps horizontally across the entire eye, drawn by dedicated muscles, moistening and protecting the surface of the eye while still allowing at least partial vision through it. Useful for a diving bird keeping its eyes open, underwater, a shark protecting its eyes during an attack, or a cat sweeping debris off the cornea without fully closing its eyes to a threat. Comparative anatomists have long treated the human plica semilunaris as the reduced remnant of that same structure, no longer capable of sweeping across the eye, no longer served by the full complement of muscle needed to move it, retained as a small fixed fold, doing a fraction of what a functioning nictitating membrane still does in a bird or a reptile today. The comparison across primates supports the same conclusion. Some lemurs and other prosimian primates retain a nictitating membrane with considerably more mobility than the human version, part of a broader pattern in which the structure appears progressively reduced, moving from prosimians toward apes and humans, consistent with a gradual loss of function rather than a structure that appeared and vanished abruptly. Exactly why the reduction happened is less firmly settled than the anatomical pattern itself. Forward-facing, high-acuity vision adapted for depth perception and manual dexterity, rather than the kind of lateral, wide-field vision more common among animals that rely heavily on a sweeping membrane for protection, is the leading explanation. Though it remains an inference built from comparative anatomy rather than something directly observed happening. What is left in the corner of a human eye is small enough that most people go their entire lives without consciously noticing it, a fold of tissue with no real job left to do, sitting exactly where a fully working eyelid used to sweep, on an eye that stopped needing it, and over evolutionary time, gradually let it shrink down to a barely there fold, rather than dispensing with it entirely. Number 1. The teeth built for a jaw you no longer have. Somewhere between the ages of 17 and 25, for a large percentage of the human population, a set of teeth attempts to arrive that frequently does not fit. Third molars, universally known as wisdom teeth, are the last teeth to develop and the last to erupt, positioned at the very back of each dental arch, and in a very large proportion of modern humans, there simply is not enough room in the jaw for them to come in properly. The result is impaction, teeth blocked from erupting normally by bone, by the neighboring second molar, or by soft tissue. And impaction is common enough that wisdom tooth extraction is among the most frequently performed surgical procedures in modern dentistry, carried out on a scale numbering in the millions of procedures a year in the United States alone. The mismatch is not a flaw in the tooth. It is a mismatch between the tooth and the jaw built to hold it, and the jaw is the part that changed. Comparative work across the hominin fossil record shows a consistent pattern of jaw and facial reduction accompanying the broader trend toward a smaller, less robust skull over the last 2 million years, closely tracked against the archaeological record of increasingly processed diets, first through the systematic use of stone tools to cut and pound food, later and more dramatically through the adoption of cooking. A landmark set of experiments led by the Harvard primatologist Richard Wrangham and colleagues, along with biomechanical modeling published by Daniel Lieberman's lab, demonstrated that cooked and mechanically processed food requires dramatically less chewing force and fewer chewing cycles than raw food, and that populations eating an industrially processed modern diet chew for a small fraction of the time and force that a population eating an unprocessed ancestral diet would require. Less mechanical stress on a developing jaw during childhood and adolescence appears to translate directly into a smaller adult jaw because bone, unlike a tooth, remains responsive to the physical forces placed on it throughout growth. The teeth themselves did not get the memo. Tooth size is under substantially more rigid genetic control and has not shrunk at anything close to the same rate as the jaw that houses it, leaving the modern human mouth in the position of a jaw built for a smaller, cooked, processed diet, still attempting to accommodate a full ancestral dental inventory, sized for a jaw that chewed far more, far harder, and far more often. Studies comparing skeletal populations across the transition to agriculture, and separately across the more recent transition to industrially processed food, document a measurable rise in impaction, and crowding tracking closely with each dietary shift, evidence that this is not a fixed condition of the species, but an ongoing, actively worsening mismatch tied directly to how recently and how thoroughly a population's diet changed. Third molars are not a leftover in the sense of a structure with no purpose left. A wisdom tooth that erupts cleanly into a sufficiently large jaw functions as an entirely ordinary, useful molar, doing exactly the same job as the two molars in front of it. What has been left behind is the jaw those teeth were built to occupy, reduced by a shift in diet that unfolded over roughly 2 million years, while the teeth kept the blueprint of an ancestor who needed every one of them and needed the full force to use them just to get through a single unprocessed meal. If you want to see more videos like this, click the video on screen now and make sure to subscribe.